Okay, here's the fourth video for chapter 11, the genetics chapter. In this chapter we're going to cover, or in this video we're going to cover incomplete dominance, codominance, multiple alleles, maybe more beyond that. We'll see how much time is left in this video. Alright, so I want you to start out with the warm-up question that's here. Taking a look at this question, pause the video, come back and check your answers for this. Okay, so for in this problem, you're crossing red snapdragons with white snapdragons. The offspring have pink flowers. So having one gene from the red and one gene from the white gives you what's called incomplete dominance. It only produces some of the red pigment, therefore it's not enough to give it the full red color, and it looks pink. If you were to cross two pink snapdragons, then what you would end up with, you can see down here, that we would have one red to two pink to one white as the phenotypic ratio in the F2 generation. All right, so what we're looking at here is incomplete dominance. In incomplete dominance, the heterozygotes have their own phenotype, and the reason for that is because the pigment that's being made, or whatever it is, whatever protein is being made, because it's only located on one of the chromosomes, it is being made half as much. Sometimes that's enough in order to give you the full effect of the dominant characteristic. Other times it's not enough to give you that full and complete effect. So we have one protein being made here. The white allele is probably a broken allele. It's probably not making any pigment color at all there because the gene has a mutation in it. It doesn't produce anything. So I don't think it's making a white pigment. I think it's making a nothing pigment. So when you have only one copy of the red gene, it's making the red pigment, but it's not making enough to fully have a red plant. Whereas in the pea plants that Gregor Mendel was looking at, in that case where they were heterozygous, they were fully purple. So sometimes you get this, sometimes you don't. In a problem that has incomplete dominance, they must tell you in the problem that the offspring have an incomplete a phenotype that's in between. So this has to be provided to you so that you know what you're working with. So yeah, red and white snapdragons produce pink snapdragons when you cross them. And the amount of pigment is production. The amount of protein pigment production is intermediate in the heterozygotes, and that's why you get that intermediate coloration. All right, let's contrast that to codominance. In codominance, they seem kind of similar, but there is a difference that I want you to be able to distinguish. In codominance, heterozygotes have a phenotype in which both the traits from both of the other homozygotes is being equally expressed. Separate, distinguishable expression, not intermediate. So for example, an example of codominance is with the blood groups. So you should be able to do blood type problems after this. In humans we have type A blood, type B blood, type AB blood, and then there's also type O. All right, so if you have type A blood, that means that you produce a protein on the surface of the red blood cells. That's an immunoglobulin, it's called, that's going to produce A. If you have B, you produce B. AB, these individuals have one gene for the A, and this is how you write it, immunoglobulin A, and one gene for the B, and so they produce both proteins on the surface of their red blood cells. So it's not just one protein being made half as much. Instead, it's both proteins being equally expressed, and that's what codominance is about. Also with blood type problems, as long as we're talking about this, we're going to get into multiple alleles here in just a second. This is an example of a trait that has more than just a dominant allele and a recessive allele. It's got an A allele, it's got a B allele, and it has an O allele. So there's three alleles that could be at a particular position at in a gene locus. And so if you run across a problem where you have multiple alleles like this, you have to be given information about what is dominant and what is recessive. So in this case, O, and this is just one you should learn, O is recessive to the other two alleles. So the only way somebody can have type O blood is if they have two O alleles, and I'm writing them as lowercase i's because they are the recessive characteristic. However, somebody that has type A blood, for example, could have two A alleles Or they may be heterozygous and have an A allele along with an O allele, which is the recessive allele. And the same thing with B. B would be being homozygous for B, or they could have a B allele and the recessive I. Okay, so those are the phenotypes for blood type here, phenotypes, and then these are the possible genotypes that go along with those. Okay, so now we're moving on to multiple alleles here. I've already explained really within that codominance problem also that blood types have multiple alleles. Not only do A and B show codominance, but O is a third allele that could be present that is a recessive to the other ones. So knowing what you know about blood type, I would like you to try this problem down here. And so pause the video, try the problem, and then come back and check your answer. All right, so taking a look, we have one parent has type AB blood, so they have one A allele and one B allele, and the other parent has type O blood, so they would have to be homozygous recessive down there. Taking a look in the Punnett square, the question is asking for what is the chance that the child has type A blood. So I've circled those in there. It's 2 out of 4, or a 1/2 chance, or you could report that answer as a 50% chance. All right, polygenic inheritance is where you would have more than one gene that's involved in a single phenotypic character. All right, so multiple genes involved in a particular trait. In that case, the phenotype is going to be an additive effect of these 2 or more genes. So, for example, human skin color is affected by at least 3 genes. So let's just think of it as having 3 genes. The more dominant alleles you have for human skin color, the darker your skin color ends up being. So you can kind of think of it as each dominant allele gives a dose of dark color. All right, take a look at interactive question 14.8. Pause this and then come back and check your answers. All right, so for this problem, we're looking at the spikeweed plant, and its height is determined by multiple genes. So we've got 3 genes here involved with the height of the spikeweed plant. There's a base height of 10 centimeters, and then you add 5 centimeters to the height for each dominant allele that you have beyond the 10 centimeters. We're starting out with a parental generation where you've got the tallest type plant crossed with this is the tallest type plant crossed with the shortest plant. And so part A is asking for what the height of the F1 plants are going to be. So the F1 plants are going to have to be heterozygous for all 3 genes, because that's the only possible combination. You have to get a big A and you got to get a little a, and so on. You have to, the only possible genotypic combination is heterozygous for all 3. So to be heterozygous for all 3, we know we get a base height of 10 centimeters plus 5 centimeters for each dominant allele, and we have 3 dominant alleles, so you're going to add 15. So the height of the F1 plants is 25 centimeters. Okay, now for part B, you're supposed to be looking at how many phenotypic classes would there be in the F2 generation. So basically it's asking how many heights would there be in the F2 generation. So we have a big problem here where you have to figure out all the possible combinations from this cross where you're selfing the F1 generation. So your cross would be big A, little b, big C, little c crossed with itself, okay? So from this cross, you have to think about what are all the possible combinations that we could get from this cross. Would it be possible to get all 6 of these alleles being dominant from this cross? Is it possible to get big A, big B, big C, big C? And yes, it is. You can set up Punnett squares for each one of them. You can look at Punnett squares for the A's, the B's, and the C's. And in each case, it's a 1 in 4 chance that you're going to get 2 dominant alleles for both of them. So it would be 1/4 times 1/4 times 1/4 would be the probability. But you can get a plant that is the full height and be 40 centimeters tall. Let's look at the other end of the spectrum. Is it possible to get a plant that only has recessive alleles from this cross? And once again, yes, it is possible. You could get little a, little b, little c, little c. It's possible to inherit that from the parent. So it is possible to get a plant that has 0 dominant alleles and is the minimum height. Is it possible to get anything in between? And the answer to that is yes, you can get anything in between. So here we're going from 6 dominant alleles down to 0 dominant alleles. So you can have 5, 4, 3, 2, and 1. All of those are possible combinations as far as the number of dominant alleles you could end up with. So how many phenotypic classes is this? 7. 7 different heights, because you do have 0 as well. So 6, 5, 4, 3, 2, 1, 0 is 7 classes, 7 different heights that you could see in that F2 generation. Okay, so this is an illustration that shows that same cross that we were looking at, where you're crossing individuals that are heterozygous for all 3 alleles and all the possible combinations that you would get. And it's also looking at the fraction of the progeny. If you went through and you figured out what is the probability of getting each one of them, more individuals would be the medium height for the spikeweed plant, or this is actually kind of referring to human skin color, but more individuals would be in the intermediate areas rather than at both extremes. All right, so there's that type of a problem, getting there. All right. You will be running across a few instances where you'll be analyzing some pedigrees. Now, I know you've seen pedigrees already in regular biology, so this is just a quick review on pedigrees. Remember that when you're looking at a pedigree, squares are males and circles are females. All right, so there's that. And then also you're looking at the various different generations as you move down the pedigree. So this would be these 2 girls are what we're kind of looking at here, and we're looking at whether they have widow's peak or not, so that growth pattern of hair. So these are the girls. Up in this next generation above them, that would be their parents. This is dad, this is mom. And then their aunts and uncles as well. And then finally up here we have both sets of grandparents for those girls. From the pedigree, we can also see whether the individual has the trait that you're following. Usually you'll shade in the individuals that have the particular trait that you're looking at, whether it's a trait that's a particular disorder, such as maybe cystic fibrosis. You might have those individuals shaded in. In this particular case, they have shaded in the individuals that have widow's peak, so we're following it. Using a pedigree, you should be able to determine whether a trait is dominant or recessive, and you should be able to go through and get practice with that. So now take a look at interactive question 14.9 for practice with pedigrees. Okay, taking a look at this pedigree, part A is asking for, is the trait that we're looking at here, which is albinism, lack of skin pigmentation, is it caused by a dominant or a recessive allele, and how can you tell? All right, so the answer to this is that it is a recessive allele. But you may never, never, never tell me that the reason it's a recessive allele is because fewer individuals have it. Recessive does not mean uncommon. There are recessive alleles that are common. There are recessive alleles that are uncommon. All right, so keep that in mind. That's not your reason here. So you do have to use the information on the pedigree in order to answer that question. So the reason we know that this is a recessive allele is because there is a set of parents here, this set of parents here, the grandparents, these guys, they don't have the disorder, but they have a child, actually 2 children, that do have the disorder. So the only way that can work is if these parents are heterozygous. And if we think in the reverse, sometimes I think that makes more sense. Let's say that it is a dominant situation. So like this guy here, he would be big A something. Then that would mean that if it's a dominant situation, that both of his parents are little a, little a. How? Little a, little a, little a, little a. How are you going to get a big A from 2 individuals that are homozygous recessive? It does not work. So this trait must be recessive. These individuals are little a, and their parents would have to be big A, little a. Both of them carrying the recessive allele and able to pass that on to their children. All right, so you're supposed to determine the genotypes of the parents in the first generation. Did that already. So that's B. This is your answer for B. And then for part C, you're supposed to determine the probable genotypes of the mates of the albino offspring. So 1, 2, and 4 we're supposed to be figuring out. All right, so some of these are a little easier than others. I'm going to do 1 last. So number 2, this male here, that's number 2, he has a child that has albinism, so little a. That child must have inherited a recessive allele from both of his parents. So mom has the disorder, dad must be a carrier and be heterozygous. All right, individual or grandson number 4. Grandson number 4 has does not have the disorder, so we know he's dominant, but he does have a dad that has the disorder. Dad only has little a's to give to him, so he must be heterozygous. All right, now mom, number 1, individual 1. She does not have the disorder, so we know she's big A something. Can we rule out the fact that she is big A little a and just happened to pass the dominant allele along to all 4 of her children? It's a possibility. It's not as likely as the likely possibility for number 1 is that she's probably big A, but based upon the information, we can't tell for sure. All right, and then part D, can you determine the genotype of son 3 in the 2nd generation? Why or why not? Okay, so son 3 over here. All right, he doesn't have any kids that have the disorder. His parents don't have the disorder, but they're both carriers. He's married or has children with a female. She's not part of this family. Being from the general populace, it's very, very likely that she's probably not carrying the allele for albinism. So in that case, you can't tell whether he's big A, or big A, little a. So the answer to that is really no, not certainly. Okay, the last point that I want to make, if you read this, cardiovascular disease is more prevalent among those whose biological or adoptive parents have cardiovascular disease. So what is the point that I'm trying to make here? All right, so if we think about this, is cardiovascular disease something that is genetic, or is it not something that is genetic? So keep in mind that there's more to phenotype than just genetics. Yes, there's probably a genetic link for some of the individuals that have cardiovascular disease. So in some cases, it's probably running in the families. But that doesn't explain why adoptive parents would also potentially be passing on cardiovascular disease to their adopted children. That they didn't do that. So keep in mind that there's also an environmental component to many phenotypes. And so if you're eating, cheeseburgers and French fries every night, that's going to be an environmental impact on your heart health. And so a lot of phenotypes have more than one component that's going into their expression. All right, that's it for these videos. Go work on those genetics problems. Do them all.